In this video, we will be reviewing how to do an siRNA transfection. So I will be taking you through the process of splitting your cells, preparing your 6-well dish to contain cover slips for our Cas-based assay, and then also preparing the transfection in the 6-well dish, and then the last step is adding the cells on top of that. So we will be moving this video into the hood so that you have a better view of how to go about this process. Splitting the cells. Okay, so I have my plate of cells and I'm ready to split the cells so that I will be able to count them and then add them to the dish after the transfection. So to begin, I will remove the media, place it in my discarding container here. I will wash it with 5 mils of HBSS. This is a buffered saline or buffered salt solution. And it serves to wash the media off of the cells because the media contains FBS, which can inhibit the trypsinization process. And the last step is adding the trypsin. So, adding the trypsin. All right. Now this is ready to go into the incubator for 5 to 10 minutes. So I could set my timer and then check on it periodically. In the meantime, I'm going to be preparing my dish for the transfection. Preparing the 6-well dish. Okay, so now I've gotten my 6-well dishes out, and I've already labeled them for our experiment. So we do need 10 of the 6 wells. We also have some cover slips here. And they have some small round glass cover slips, and they've been labeled with the letter R. And so the goal here is to take our glass cover slips and to put 1 cover slip into each well for numbers 3 through 10. We do not need one for 1 or 2, but we're just going to start here with number 3, and we're going to add the glass cover slips. Now, working with these glass cover slips can be a little bit tricky. And just keep in mind that everything here has been autoclaved and is sterilized and ready to go. I'm going to clean this with some ethanol here real quick. And we're going to use this needle to just assist us in picking up this glass cover slip. And so what you can do with this is you can use this needle to kind of pick up the cover slip and get it up on its side. And then with your other hand, you can come in with some tweezers. And I have the tweezers here and ethanol over here. And then, sorry, I just dropped it. With your other hand, you can come and pick it up. Okay, I'm holding the phone with one hand, so it's a little tricky for me to show you, but that's the goal here. And what you're going to do is you're going to pick up one of these cover slips. And we'll see if I can do this with one hand or not. And if not, I'll have to show you when you come to the lab. I picked one up, but unfortunately it's not, the label is kind of coming off. But you can see that it does have an R and it has a direction to it, right? So what I'm going to do is I'm going to take this 1 cover slip, but I want the R to face me so that I'm able to read it. I'm going to remove the lid from my plate, and then I'm going to add this cover slip inside of the well on the bottom. And the goal of this is to have it so that the R is facing up towards me so that when I look down at my well, I can see the letter R and it's facing the correct direction. And what that's going to do is that at the end, when I add my cells on top of this cover slip, the cells will be growing on the side of the cover slip that has the R that faces in the correct direction. And then when I'm coming back to collect my cells during the caspase assay, then I will treat the cells with the caspase reagents. The cells that are growing on the cover slip will be processing the caspase reagent, and then I will collect the cells using tweezers. I'll pick them up, and then I'm going to put them on a microscope slide. So that's the goal with this. So we're kind of doing two things at once. We're treating enough cells in our 6-well dish that we have cells growing on our cover slip that we're going to use for the caspase assay. We're going to put those onto a microscope slide. The rest of the cells that are growing in that 6-well dish are enough for us to collect RNA, and we will use that RNA, process that, we're going to purify it, make the cDNA, and then we're going to do the qPCR. Okay, when I'm done with my tweezers, I can pick them up and put them back into that 50 ml tube, and it is full of 70% ethanol, so you want to make sure you got the clean one. Just be careful. There are people that do mouse work in here. And so there's going to be some that look like this. And so that's kind of some tissue, some kind of yucky stuff down there. So make sure that you're not grabbing that one. We certainly don't want that in our tissue culture. But we want to make sure we have a nice clean one and that we have our tweezers clean and ready to go. Once you have the cover slips in numbers 3, 4, 5, 6, 7, 8, 9, and 10, one cover slip in each one of these wells, then we are ready to get our transfection reagents ready to go.
Preparing the transfection.
Okay, so now we're ready to begin getting our reagents into our 6-well dish for our transfection. So the first thing I'm going to be doing is adding OptiImmune. The OptiImmune is similar to media with the pH properties and buffering system that it has. It, however, does not have FBS or serum. So it's a serum-free or serum-reduced media, depending on what you purchase. And that can assist in the development of the lipids forming around your DNA or siRNA that you're trying to transfect. And it makes the transfection work a little bit easier. So my first step is to add OptiImmune to each of the wells. And so I will be adding 500 microliters to each well. And I get my 1,000 microliter pipette, my 1,000 microliter tip. I have it set to 500. Now just remember when you're using this tip to move slowly with your thumb as you're releasing. If you go too fast, I don't know if you can see it. I think my hand is in the way. If you go too fast, you'll see it jumps, and it didn't do it this time, probably because the pipette's already wet. But that's when you can get those air bubbles. So just move a little bit slower with your thumb, and you'll be better off. And I'm actually going to just go ahead and use the same tip. I'm going to add 500 microliters to each well. I'm not sure if you can see that. All right, so I've completed one of my dishes. And I'm going to move on to the second dish. So I'm just taking the 500 microliters and I'm just moving from well to well. Okay, I'm going to throw my tip away. And now I have added 500 microliters into wells numbers 1 through 10. They all contain the same. I'll go ahead and add my cap back onto my OptiImmune to keep it nice and sterile. I put that right there. All right, the next step is to add the siRNA to each well. And so we have them labeled so we know which ones get no SI and which ones get SI control and which ones get SI for our target gene. So you will have a tube for SI control and a tube for your SI for target gene. In my case, I'm using SI active. All right. I need to get the correct tips over here. So with my calculations, I found that I needed 7.5 microliters. So I'm going to get my 2 to 20 microliter pipette. And I'm going to set it to 7.5. So it's ready to go. And I can get my SI control. I don't need to put anything into wells 1 or 2 because they do not have an SI. They're strictly for controls. My SI control, remember the SI control is a scrambled siRNA. So it does have siRNA, but it has a scrambled sequence that should not recognize any genes. And so it is a control for having this short, double-stranded small piece of RNA in the cell, but it should not knock down any genes. And so that SI control is going to go into numbers 3, 4, 5, and 6. So I will begin doing this. 7.5 microliters is a very small amount. I want to make sure that I'm checking my tip that I have collected 7.5 microliters. It's kind of easy to mess that up. I will add it to my well. Let me just, and here so you can see, and I'm going to pipette up and down, maybe 2 or 3 times. So there we go. I did 1. And then I will continue to do the other 3. So because I might not use all of my SI control in one day, I want to keep it sterile. So I am going to change my tip. Okay, I am finished with my SI control. And keep in mind that every time I added it, I was adding it from the same tube. And I added 7.5 microliters into wells 3, 4, 5, and 6. Now I'm ready to add my SI axle to the wells. Right here, I have them labeled SI target gene. I could have labeled it SI axle because that's the gene that you guys picked for your project, right? So when we did our calculations, it was the same, 7.5 microliters. And I'm going to be adding it to my well. And I need to repeat that 3 more times. Okay. I've added SI axle to well 7, 8, 9, and 10. So I took that all from the same tube. I put 7.5 microliters into each one of these. So I have now diluted my siRNA in the OptiImmune. I can give it a little shake just to help mix that up. And now I'm ready to add my RNAi Max. So I have a tube of RNAi Max. And I'm actually going to be adding the same thing to each well. So this is going to be 9 microliters for the EW8 cells. Keep in mind if you have RH41, they are more sensitive to the RNAi Max, so you need to use less. In this case, I'm going to go ahead and use the same tip because I know I'm going to run out of this RNAi Max. This time, I do not add it to number 1 because this is control with nothing in it. Number 2 is lipo only. So I will add that to the lipo only. I can set my lid down there if I'd like to. Okay, I'm going to keep adding this to each one of the wells. So I added that to well 3, 4, 5, 6. Okay, and I pipetted up and down 2 or 3 times to make sure that the RNAi Max went into the media. So now I've added the RNAi Max to all of numbers 1 through 6. I will repeat for the remaining wells. So it's still RNAi Max. I'm still working on the same tube. I am removing RNAi Max. And I'm putting it into numbers. Oh, I lost my tip. 8, 9, and 10. Okay. I will remove my tip and put my pipette over here. Okay. At this moment, we have our wells. We've added our cover slips. We have 500 microliters of OptiMEM. We have put SI control into the wells that say SI control. We have put SI axle in our SI target gene wells, so 7 through 10. Even though we've labeled minus drug and plus drug, we are not adding the drug today. We are going to add that 24 hours before we collect. So at this moment, we have the OptiMEM, the SI control, and the lipofectamine RNAi Max. So I'm just mixing it because I want all of that siRNA and lipofectamine to interact with each other. I want it to be mixed. At this time, we're going to do a incubation time of 10 to 20 minutes, and that will allow the complexes to form within the well. And at this moment, we do not have any cells here yet. So the complexes are forming where the lipofectamine would be surrounding the siRNA. And once that has had time to incubate and those complexes can form, then we will add the cells on top of it. And you can just leave your dishes here in the hood. They do not need to go into the incubator for this step. It just incubates in the hood for 10 to 20 minutes.
Resuspending the cells.
Now we're going to address our cells, which we're trypsinizing in the incubator. So we have collected our cells, placed them into the dish, into the hood, and now we're going to resuspend. So we have trypsin in the dish, the cells are trypsinized, and I'm going to be add 4 microliters or 4 mils of media. I added to my plate, and I pipette up, and I wash plates. Just to catch all the cells that are stuck on the other side of the plate. For the purposes of culturing the dish, I bring all of the media up into my pipette. I will leave 1 mil in my plate, half a mil to a mil. I have actually already labeled a 1.5 mil tube. I'm going to put just a little bit in here so I can do some counting with the hemocytometer. So I'm just going to put that into a 1.5 mil tube. And I've already labeled a 15 mil tube. And I am going to place the rest of the cells into this 15 mil tube. So place the cap back on and then placing it back over here. And I can discard of this. For this, I'm going to add 10 mils of media. Into my 10 centimeter dish. Now that 10 centimeter dish has media and a little bit of cells, and that dish can actually go back into the incubator, and within a couple of days that dish would be full of cells again, and if we needed to do another experiment, we could. Okay, so with my cells that are in this 1.5 mil tube, what I would do is I would do the dilution by adding some trypan blue, some PBS or HBSS to dilute it, and then some cells. So like we've done before, usually we do a 1 to 10 dilution and prepare that in a new 1.5 mil tube. use 10 microliters of your dilution to count the cells on the hemocytometer.
Plating the cells.
Okay, now that I've counted my cells on the hemocytometer, I'm ready to prepare my cells so that I can add 5 times 10 to the 5 cells in each well. So I have already labeled a 15 ml tube. I've labeled it EWA. I went ahead and just wrote 5 times 10 to the 5 cells per mil so that I could distinguish it from my other 15 ml tube, which would look very similar if I just put EWA. So that helps me to distinguish between the two. This is what I'm going to be making before and mixing so that I can add the cells into each one of these wells. Okay, so I'm going to just put my lid here for the moment. And my calculations said I had 1.67 times 10 to the 6 cells per mil. And in order to make 12 mils of a solution where each mil contains 5 times 10 to the 5 cells, I have calculated that I need 3.59 mils of cells. So I'm going to get my pipette. And I'm going to take my stock solution of EWA cells. I want to pipette up and down a couple times. See if you guys can see that. Because the cells would be stuck kind of at the bottom. So gravity works pretty fast and the cells settle to the bottom. So I want to resuspend them before I add them to the new mixture. So I'm going to get 3.6. I'm just going to go ahead and estimate close to 3.59. And then I'm going to grab my new tube and I'm going to add my cells to that. I have calculated that I need 8.4 mils of media. So I'm going to get a 10 mil pipette. And I'm going to grab 8.4 mils. And I'm going to add it to the mixture. All right. My next step is to add 1 mil to each of these wells. I will take my pipette and I will very carefully pipette up and down to mix the cells. I really have to watch the top of this media. I do not want it to go up into the red zone where those little red lines are before the filter, because when that happens, the filter gets full and that actually clogs up the filter that's in the pipette, and it'll stop working. So I gotta be careful. All right, so I'm almost ready there. Now I could take either a 10 or 5 mil pipette, and I could release 1 mil into each well if I felt confident doing that. Or alternatively, if you're not too steady with the pipette yet, you can use the 1 mil pipette. Be sure to move it to 1 mil. So I have it dialed in to 1 mil. And adding a pipette tip. Let me just rearrange my stuff so I'm ready to go. Okay. So I can move my lid to the side. Let's see if you guys can see that. I will add 1 mil of cells to each well. I can use the same tip and just keep adding it to each well. I finished one of my 6 wells already. I'm going to put the lid on, move it to the side. Now I'm ready to work on the next one. Okay. So now I have my dish. It contains my transfection and it contains my cells now. So I will just swirl it gently like this. You have to be careful about going in circles because sometimes the cells end up all in the center. So a lot of times I just go back and forth. I let it settle a little bit and then I go side to side. And I might do this a couple times, but eventually they will get mixed up. And I'm going to do that to both of these. I'll probably do it a couple times as I'm walking over to the incubator. I can stack them together like this. And now I'm ready to carry them to the incubator. And then they will incubate for 2 days. And that will give the cells time to grow, but also gives the siRNA time to kind of take effect. So it takes a few days to see the effects of the siRNA, especially when you're looking at some downstream signaling. So we'll give it 2 days, then we'll treat with drug. So we will be adding the drug directly onto our plate. And then the next day we will collect for RNA, but also for the caspase acid.
Treating with drug.
Okay, so our cells have incubated for 2 days, and we have taken them out of the incubator and put them back into our tissue culture hood. And now I have my drug, and this is the concentrated drug, so it's our stock, and we need to make a dilution of it before we add our drug to our cells. So based on the concentration of the drug that was given to you, then you do your calculations. So in my case, I need to do a 1 to 10 dilution. I have decided, let's just make 50 microliters. So I'm going to take 45 microliters. And you'll have to discuss this with your teacher before you get started to make sure that you're using the right dilution. But I'm going to take 40 microliters of DMSO, and I'm going to add it to my new tube. And then to do a 1 to 5 dilution, then I need to take 5 microliters. So I have that set, and let me look at that. So there we go. Okay, so I'm going to take my stock drug, which I've already mixed up, so it's mixed up pretty good. You do want to make sure everything's mixed when you're doing this. I'm going to pick up 5 microliters, and I'm going to add it to my new tube, so it's 45 microliters of DMSO, 5 microliters of the stock drug. And that gave us a 1 to 10 dilution. Okay, so I'm just going to vortex this over here on the side. Mix it. I'm kind of a little bit mix happy when it comes to the drugs. I want to make sure they're all mixed up. Okay, so this is my new drug. I have a concentration that has been diluted, and it's ready to be added to the wells. So just based off of your calculation, you will determine the microliter amounts. So don't rely too heavily on this video for the microliter amounts. It will depend on your concentration for your drug. Now what I'm going to do is in the wells that say minus drug, I'm going to add DMSO. And I add the same microliter amount that I will add the drug into the wells that have drug. So if my calculation said I need to add 1.5 microliters of drug to each well, then I will add the same amount of DMSO to the controls. So I'm going to take my DMSO tube. I'm going to add 1.5 microliters to the SI control minus drug well number 3. So I just pipe that up and down a couple times. You will need to change your tip on this one because you are dipping into your wells that contain cells now. And then you'll just continue to do this. So number 3, number 4, Number 7. Oops, I just dropped it. And number 8. Okay, so I've added all of my minus drugs. The minus drugs are just DMSO. Okay, so I can mix that around a little bit. I don't want to mix too heavily now that my cells are grown. And then I'm going to take my diluted drug. I'm adding 1.5 microliters in my case to the wells that say drug. So in this case, it's number 5. Number 6. I'm just pipetting up and down inside the media a couple times. Number 9. Number 10. So you do want to pipette up and down a couple times just to make sure that drug is getting out, and always be visually looking at your tip to make sure that liquid is moving in the way it's supposed to be moving. Okay, so that's it. I've treated my cells with the drug, and I can just shake them gently back and forth, but real gently, especially since you have cells growing there. You do not want them to get disturbed. You don't want them to come off of those cover slips. So now that I've treated the cells with the drug, they will go back in the 37-degree incubator for another 24 hours. After that time, we will be coming in to collect the cells for RNA lysis, but also for the caspase assay. And that's where we will be removing the cover slips after we've done treatment of the cells with the caspase reagents. We will remove the cover slips, put them on a microscope slide, and then with the cells that remain in here, we will be collecting them for RNA purification.
Thanks for watching. Feel free to go back through the video and catch anything if you missed it.